Hello, good morning, students. Last time we had studied about enzymes. Today we will study mechanism of enzyme catalysis. We know the important role of enzyme is act as a catalyst in biochemical reactions in physiological processes. So the action of enzyme on substrate is described as lock and key mechanism. Accordingly, enzyme has active site on its surface. This we had studied last time. So see this structure of enzyme. This is yellow colored part, and this is the space where the key can be put, like key and lock type of arrangement. This site is called as a active site. So enzyme has active site on its surface. A substrate molecule can attach to the active site only if it has the right size and the shape. Say this substrate molecule, it is a green colored molecule is shown, and the active site of enzyme, if the active site size and the shape of substrate that matches, then and then only this reaction is possible. And therefore we had studied last time that these reactions are very specific reactions. Enzyme catalyzed reactions are very specific or highly specific. So when the active site of substrate is held in a correct orientation to react and forms the product of reaction, the products leave the active site. The enzyme is then ready to act as a catalyst again. See this figure. In this figure, this is a substrate, green colored molecule. Yellow colored molecule is enzyme. Enzyme has active site. This is a lock and key type of arrangement. If the size, shape, and the orientation that matches, then and then this reaction is possible. When this substrate binds with the enzyme through this active site, then the complex is formed. This is called as an enzyme substrate complex. When the reaction takes place, the product is formed, and again product is separated from this enzyme active site. So this enzyme is again ready to act as a catalyst again for the next reactant, next substrate. The formation of enzyme substrate complex has a very low activation energy, means energy required to bind these two molecules is very less, and that is how the rate of reaction is very high. Some enzymes are so efficient that one enzyme molecule can catalyze the reaction of 10,000 substrate molecules in 1 second. So reaction speed is very fast. So these enzymes are highly efficient. The rate of reaction is very high. Here one example is given. The one enzyme molecule can catalyze the reaction of 10,000 substrate molecules in 1 second. So several enzymes have been isolated from the organism, such as bacteria, purified and catalyzed. Amino acid sequence of many of them have been determined. In many industrial processes, specific reactions are carried out by the use of enzymes extracted from organism. and also by the use of new enzymes made using genetic engineering. So this branch is very useful nowadays to develop the catalyst enzymes and the speed up the reaction. Some examples of industrial applications of enzymes are given here. One is the conversion of glucose to sweet testing fructose using glucose isomerase. This reaction is carried out by using enzyme glucose isomerase. Then second is mechanism of new antibiotics using penicillin G acylase. So enzyme used here is penicillin G acylase. Then manufacture of laundry detergents by using protease enzyme. Then manufacture of ester used in cosmetics using genetically engineered enzyme. So this is very important application of these enzymes, and that is about the mechanism of enzyme catalysis reaction. Thank you.